Hello everyone, good afternoon. Welcome to Conway Hall in quarantine, Thinking on Sunday. We are once again broadcasting from the Conway Hall shed and various other places. It's good to have you all here. I hope you're all keeping well. For those of you who've not attended either a live Conway Hall event or an online one before, Conway Hall exists as a charity to spread ethical thought, philosophy, and ask questions about the world. And Thinking on Sunday is the latest version of the Sunday talks that the Conway Hall Ethical Society and its forebears have been running for a long time. Just so you know, how we do things is we will introduce our speaker in a moment. They'll speak for about 45 minutes or so, and then we'll have a Q&A. That will be done by, if you don't mind using the chat box in Zoom, which is hopefully at the bottom of your screen. Type your question in there or put your hand up if you wish to. And then we can either open up your microphone and you can ask the question live. Don't worry, we won't be able to see you, only hear you. Or we can read out your question, but do it live. It's a lot more fun, and everyone gets pretty fed up with my voice pretty quickly. If you are tweeting, we have a hashtag, which is pretty straightforward, hashtag Thinking on Sunday. If you want to share your thoughts on the talk while you're listening to it, and look out for more events at Conway Hall. We've got loads coming up live. And if you are feeling comfortable with it, with doing this, let us know where you're watching or listening to us from today, because obviously normally we're in central London, but we are broadcasting now to the world. This Sunday afternoon's talk is Dr. Kat Arney. Kat is a broadcaster, science communicator and storyteller, and author. She's a doctor working in the field of cancer research. And cancer research is what she will be talking about this afternoon from her latest book, Rebel Cell, which is new ways of looking at cancer. So hopefully there'll be a peaceful transition to Dr. Arney. Please welcome Kat Arney to Conway Hall Thinking on Sunday online. Thank you.
Thank you very, very much. I hope everyone can hear me. And also I am going to try and share my screen so that people can see my slides. So hopefully everyone can see that as well. Get myself out of the way. There we go. I want to say thank you so much to the Conway Hall team for having me. Conway Hall is a really important part of my life. I've been a member of the Conway Hall Ethical Society for several years now, and many people who are my good friends are involved in running it. And I just think it's so important that we all keep something like Conway Hall going through this time, because it's more than just a building. It's a place, an ethos. It is people who can come together, who can exchange ideas, who can talk, who can think. It's a wonderful home for free thinking, for secularism, for science, for discussions. I think it's just an incredibly powerful place. So I'd urge all of you, if you're not already a member of Conway Hall, to join, to donate if you can, because yeah, it's been a rough year, very rough year, and I think it's going to continue to be difficult. And I know that the team and the trustees are just doing an incredible job to try and work out how to move forward in an events-based business when events are very tricky to hold. So it's really fantastic to be part of an online event like this. It's very special. I feel very sad that I can't be up on the stage because it's just such a glorious building to be in. But also it is very nice that I can do a talk to all of you while I'm wearing my slippers and not having to wear high heels. So, you know, swings and roundabouts as we go. But I'm here. My name's Dr. Kat Arney. I am a science writer. I'm a broadcaster. My background as a scientist, I trained in genetics and developmental genetics. And then I spent 12 years at Cancer Research UK in the science comms team. And then 4 years ago, I brought out my first book, Herding Hemingway's Cats, which is all about how our genes work, you know, trying to understand how does this string of code, this string of letters, make us who we are? How do you go from one set of DNA to building a baby? That's just the most incredible question. I think it's the most fascinating thing in all of science, really. You know, you see Brian Cox doing all this stuff about the stars and the universe. It's like, How do you make a baby from one cell and one set of instructions? I think that's absolutely incredible. So my first book was really exploring that idea about how do our genes work? How do they make us who we are? And then for this book, Rebel Cell, which came out in the UK in August, great timing, launching a book about a life-threatening disease in a life-threatening pandemic. Great timing on my part. But this book is all really exploring some of the themes that I came across while I was working at Cancer Research UK, but also since then, the advances that we've seen in the world of cancer genetics, in genomics, in our understanding of this disease, and realizing that there was time to tell a new story about what cancer is, about the ways that we treat it, about what we're doing well, what we're maybe not doing so well, what the way forward needs to be, and how do we almost come to a new view of cancer that is really starting to emerge in the scientific and the medical worlds, but I think hasn't really filtered through to the public so much. So that's what I wanted to try and do with my book. And that's what I'm going to try and do in the short amount of time that I have to talk to you today. So I'm going to explore a bit about where did cancer come from, what makes it so difficult to treat, and then also some new ideas emerging about how we can treat particularly advanced cancers more effectively. Now, as a little warning, content notes, at one point in the talk, I do show some medical images. Some people might find that a bit upsetting. There's nothing really, really gory. It's just, I will warn people. So if you're not very happy with seeing medical images, then just be warned about that. So where are we going to start? Well, I want to start with the good news. So we know that today in the UK, half of all people who are diagnosed with cancer today will survive their disease for 10 years or more. And that is really incredible progress. That's a figure that's doubled in my own lifetime. And so when I was writing this book, what I really didn't want to do is make people think that we've made no progress, all the effort, all the money, all the energy, the resources that we've put into cancer research and cancer treatment has just been a waste of time, because it absolutely has not. We've made significant progress in how we diagnose many cancers, in how we treat many cancers, things like in treating childhood cancers, in treating bowel cancers, breast cancers, particularly cancers that we find at an early stage before they've really started to spread dramatically through the body. We're getting really good at finding and treating those cancers. So, in some ways it's a glass is half full. But what I really wanted to explore in this book is how do we get that glass full to the top, if indeed we can? Because what I came to realize is that a lot of the progress, the treatments, these in some cases incredibly expensive wonder drugs that we read about in the newspapers, they are really only adding droplets into this glass. And I think that we need a new understanding, a new way of thinking about cancer if we're to really start filling that up. Because this is a disease that touches us all. It's a disease that either we may have suffered ourselves, people close to us, people have gone through it, we've lost people that we love. It's just something that we all care about so much. And as I came to understand, is something that is really intrinsically linked to our humanity and also linked to our place in life on Earth as well. So one of the real big myths that I wanted to dispel in writing Rebel Cell was that this idea that seems to have cropped up, and we sometimes hear about it, this idea that cancer it's just a human disease, it's our problem, and also that it's a modern disease, that somehow our modern lifestyles, the things that we do, our terrible toxic environments are responsible for cancer. And if we could just go back to some kind of pure, wonderful world where everything was healthy, then there would be no cancer. That this is a modern problem and it's a human problem. And that is just categorically not true. So to address the human side of things, we find cancer across every branch of the tree of life. I cannot stress this enough. Cancer is not just a human disease. So there's a book that I leaned on quite heavily writing mine. This is a sort of scientific textbook. It's called The Ecology and Evolution of Cancer. It's by Beata Ujvari and her colleagues. And in here, they have a table. It's like 20 pages of close type text outlining all the species that we know of so far where there are examples of cancer that have been documented. So we've got things like lizards, Bell's dab lizard, the golden tegu, the Nyassa lovebird, we have parakeets, we have quails, we have swans, we have the glossy ibis, fin whales, ringtail cats, bisons, buffalo, the eastern barred bandicoot, the potto. I don't even know what a potto is, but it can get cancer. So this really does show that everywhere on the tree of life can get cancer, and that includes humans. So this is not just a human disease. This is a disease of life. And one of the wonderful examples, so on the right, this is an organism called a hydra. And these are very, very tiny animals. They basically live in water. They're little more than a tube with tentacles on the top. They just have 3 different types of cells, kind of inside, outside, middle, if you like. But in 2014, some researchers published a paper showing that they had found a completely naturally occurring cancer in a hydra. So this wasn't they'd done anything funny to it. They hadn't genetically manipulated it. They hadn't given it any weird chemicals. This tumor had spontaneously turned up in something as simple and tiny as a hydra. And so we find cancer across the whole of the tree of life. And this suggests that it's a deep biological phenomenon that goes way, way back into deep evolutionary time. And this is also true when we consider back in history the kinds of organisms that we know had cancer. There's evidence of tumors in dinosaur fossils. The very week that my book came out in the UK, there was a paper published showing that a dinosaur fossil had been found from 77 million years ago with an osteosarcoma, a bone tumor. Particularly duck-billed dinosaurs called hadrosaurs. There seems to be a relatively high number of cancers in fossils from that particular species. This was work done by some American researchers who went around all the natural history museums in America with a scanner and said, can we pop your dinosaurs in the scanner? And they were like, I mean, I guess so, because no one had ever really looked for examples of cancer in ancient history. And one of the oldest examples, there's a 240 million year old turtle fossil that's been found with a tumor. So this is a deep ancient disease. And in human populations, in ancient human populations all around the world, when we look for evidence of cancer, we find it. And there's a bit of an interesting argument here because the incidences of cancer are still relatively rare. There's at least 250 documented, well-documented examples of human remains found with tumors in many cases going back many tens of thousands of years. But pretty much everywhere you look, you start to see evidence of cancer in the human records. So, for example, we find what would be considered to be very rare childhood tumors. I think the fact that we can find them at all tells us that cancer has always been with us. Unfortunately, we don't know how prevalent it was in antiquity because you don't kind of dig up nice age-matched epidemiological series of human remains. You get what you get and you have to figure it out. What we do know is that the incidence of cancer does significantly go up after our 6th or 7th decade. So the rate at which your chances of getting cancer do go up once you reach your 60s. And I'll talk a little bit about why that might be later. And certainly in historical times, your chances of living into your 60s were not as good as they are now, let's put it that way. So I think there's one reason why we do certainly see more incidence of cancer in our modern populations, but it was there in the past. And as we know from looking at animal species and looking at the deep time fossil record, it always has been with us. And that's because, as I really came to understand while writing the book, cancer is the price that we pay for being multicellular organisms. Cancer is the price of life. Because as soon as you have an organism, an animal that is made up of multiple cells, you have to have kind of a social contract. So cells have to know what they're doing. They have to multiply when they're needed and not when they're not needed. If they're damaged, they need to die. They need to not consume more resources than is appropriate for them. They need to not pollute the environment around them. And they need to do the job that they're meant to do. And since the very evolution of multicellular organisms, like a billion years ago, we have seen this sort of multicellular social contract emerge. So there are cells in organisms called germ cells, and these are the cells that become eggs and sperm. They go on to the next generation, and those are very precious protected cells. And then the rest of the body, the rest of the multicellular body, is what we call the soma, somatic cells. And this is the kind of the pact of multicellularity, is that you preserve the germ cells. They're the ones that go on to make the next generation. And the soma kind of does the dirty work of getting you through life, you know, enabling you to get to a point where you can reproduce, to pass your genes on. And as in any society, there are individuals, and certainly in the case of the cellular society, there are cells that can break those rules. They can break that social contract that keeps the body healthy, and they emerge as cheats. And so this concept of cancer as cheating cells emerging in a society of cells, and they cheat because they have genetic changes, they have faults that enable them to thrive in the society that they're in. These cheats can prosper in the society of cells, and they emerge as cancer. And so this is kind of an interesting way of viewing cancer, because I think we almost think about it as a sort of an incident, something separate, but these are our cells that have gone rogue within us. They've worked out how to cheat the rules. They multiply more than they should. They don't die when they're meant to. They stop doing the jobs they're meant to do, and they start wandering off around the body. They pollute the environment around them. They take more resources than they need. They are cheats. And this has always and always will happen in multicellular bodies, in multicellular societies. So this is why cancer is such a deep, deep biological phenomenon. It's not just restricted to humans. It's not just restricted to modernity. It's a deep biological and almost inevitable phenomenon. So this led me to this was a bit of a late night after a couple of scotches sort of thought. It's like, if there are aliens, and if there are like multicellular aliens, like, could they get cancer? Like, probably. We don't know what their rate of incidence of cancer would be like, just as there are different rates of incidence of cancer across the animal kingdom. Actually, humans are not the worst. Humans are somewhere in the middle in terms of how likely we are to get cancer in our lifetime. You have very big, long-lived animals like whales, like elephants, bats, weirdly, small, long-lived animals. Down one end, they're very, very cancer resistant. At the other end, you have things like very small rodents are extraordinarily cancer prone, and humans are kind of somewhere in the middle. We do things that don't help us. We know that things like our behavior, particularly tobacco smoking, does boost that incidence up. But broadly, if you take all of that out, we're somewhere in the middle. So it's a nice thought experiment. Maybe you can think about this later, is where would aliens be in the spectrum of cancer incidence? Because my bet is that they would have it. So something to think about. So when I was working at Cancer Research UK, I was in the comms team for a long time, and we would have to do things like write a piece saying, what is cancer? So the basic explainer about what is cancer. And I would always sit down and start in the same way. You know, cancer is a disease that starts when a cell picks up genetic mutations and it multiplies out of control. And this kind of makes sense in what I've just told you about cancer emerging out of this sort of multicellular society, because if a cancer cell has picked up genetic changes, genetic faults, we call them mutations, it makes it able to cheat. It enables it to break the rules and cheat the cells that are around it, and to prosper and thrive in that situation. And this has led to the sort of what I like to call it's technically called the somatic mutation theory of cancer. I like to call it genetic bingo, where if you've picked up enough genetic changes, enough mutations, you can become a cancer cell. You've picked up enough damage that enables you to really get going, overcome the cells around you, and start to thrive, to spread through the body. So this has been a model that we've understood cancer by for a very long time, certainly for many, many years. And we have very much focused on what is it in cancer cells that has gone wrong. So there's been a huge amount of effort generating shopping lists of mutations that we find in tumors. You take a tumor, a sample from a tumor, you mash it up, you throw it through a DNA sequencer, and you get this big list of all the genetic changes that are in there. And you say, right, that's it. It's these genetic changes that are making this cell multiply out of control. And that must be what has enabled it to prosper, to grow, to thrive within the body. Right, good. That's how we understand where cancer came from. Now, there's one little problem with this, is that it assumes that normal cells don't have any of these problems. And unfortunately, recently, this was proven not to be the case. So some researchers who are at the Sanger Institute in Cambridge, go back, there we go. Some researchers at the Sanger Institute in Cambridge, they did a really, in some ways it seems a very simple experiment, but I think it's a very clever experiment in that they said, well, we come at the problem of cancer by just studying tumors. Like we study the thing that's gone wrong. And as scientists, that's what we do. We focus on the thing that's gone wrong. We don't often think about what makes us healthy. Let's look at healthy, normal tissue and try and figure out, well, what's gone right. And so what they did is they got samples of skin. So they were looking for parts of the body where they could expect to find like some damage, these cells had seen some stuff, but that was easily accessible, and that then they could actually look at healthy, normal tissue and see if there were genetic changes in there. And they realized a great place to look for this was actually the eyelid. And there's a certain type of surgery that people can have if you have eyelids that sort of start to droop and interfere with your vision. It's a type of surgery, it just removes a little flap of skin from the eyelid, and normally that would just go in the bin. And so they went to the local surgeons who were doing this type of operation, and they said, can we ask your patients if they don't mind us using their skin? And the patients were like, yep, that's fine. We don't need this. And so they got these little flaps of skin from people at various ages, and they chopped them into tiny, tiny, tiny little sections and put each one of those through a DNA sequencing machine, because now it's become so cheap and so quick and so sensitive that you can really find out what kinds of genetic changes are in a really tiny sample of cells. And what they found was absolutely staggering, was that in this completely normal looking skin, it was a patchwork of mutation. There were patches of cells here with these changes, patches of cells there with these changes, some of which were changes that if they had found them in a cancer, they would have classed them as cancer mutations responsible for the cancer. And then they were like, well, this is very strange. Let's look at a different tissue. And they looked at the esophagus. This is the tube connecting the mouth to the stomach. And in this case, they went and found people who had passed away in road traffic accidents and got consent from their families to take small samples of esophageal tissue. And they did the same thing. They chopped it into tiny, tiny, tiny sections and put it through the DNA sequencer. And they weren't expecting to see as much as the skin. Your skin is exposed to ultraviolet light. Maybe, yeah, it can pick up some changes. The esophagus, it's on the inside. Like, what's the worst that's going to happen to it? But they were absolutely staggered to find that these are three different types of samples. These are from people in their 20s, people in their 50s, people in their 70s. That even the esophagus is a patchwork of mutation. Each one of these circles represents a little patch of cells with genetic changes in there. Again, that if we found them in a cancer, we would say that that is a cancer driving gene fault. And this is normal tissue. There's no sign of cancer or any problems here. And you can see by the time you get to your 70s, you've seen some stuff. Stuff is really going on. And they've done similar research in looking at the blood. Again, loads and loads of different groups of cells. We call them clones with all sorts of different mutations in the blood. In the endometrium, the lining of the womb, by the time a woman is in her 60s or 70s, like it's pretty much all mutant in there. So, you know, ladies, we can look forward to that. But we are a patchwork of mutation as we age. So all our cells are a bit sad. They've all seen some stuff. They've all picked up DNA damage. And so when we have this very simple definition of cancer as it starts when a cell picks up genetic mutations and multiplies out of control, these patches of completely normal tissue are cells that have picked up genetic mutations and are multiplying. So this really begs the question, like, what is going on here? Because if all our cells are mutated, then what does that mean for cancer? And it's important at this point that we remember that cancer is actually incredibly rare. And this is a hard thing to get your head around, because we know that in the population, on a population level, cancer is incredibly common. So 1 in 2 people will get cancer at some point in their lifetime. That's the latest figures. So you're probably looking at me at this point and going, what do you mean it's rare? Like, that is not rare. But it's rare on the level of your body. You are made of trillions of cells that multiply millions, billions of times in your lifetime. That it's just phenomenal odds. And given the number of mutations that you pick up in your life, the number of cells that you have, the number of divisions that they make, you will probably, if you're lucky, get maybe 1, maybe 2 cancers in your entire lifetime. That's odds of, I think it's 1 in 10 with 14 zeros after it. It's more than the stars in the Milky Way by a long shot. Like, on a personal, individual level, cancer is incredibly rare. But we know that cancer does happen, and we know that cancer is likely to happen. But we don't really focus on why it doesn't. So out of all those trillions and millions and billions of cells, what's actually helping to keep our tissues healthy through most of our lifetime, up until our 6th or 7th decade for most of us? You know, we don't really think about what keeps us well. We tend to only think about like what makes us sick. So there's some really interesting areas coming, I think, in understanding what prevents cancer. How can we push that boundary away to keep our tissues healthy, to keep it under control for as long as possible through our lifetimes? So I think that's an interesting question. The second interesting question is that given that we know that our bodies are a patchwork of mutation, as I say, and, you know, if every cell, we can say, probably has some damage that if we found it in a cancer, we would say that was a cancer mutation. Like, what is going on here? And so there is, we can define now, we think that there's a key kind of tipping point that turns a sad cell into a bad cell. So by a certain age, you know, all your cells are a little bit sad. They've all seen some stuff. But very, very, very few of them will turn into a cancer cell, will become a bad cell. So what is that tipping point? What do we know about it? And it's increasingly becoming clear that it's actually not just at the level of the genes, not just at the level of specific changes in DNA, but a broader level of the chromosomes. Now, this picture, it shows the 23 pairs of human chromosomes. So you have that in all of your cells, you have 23 pairs of chromosomes. Got 1/2 of each pair from your mum, 1/2 of each pair from your dad, way back when. But in all our cells, we have 23 pairs of chromosomes. And our genes are, you know, strung through each of these chromosomes. Each chromosome is just a long piece of DNA with lots of genes lined up on it. Now, a normal human cell, the chromosomes look like this. You have nice, neat pairs, and we can paint them in different colors so we know which ones we've got, which ones we're dealing with. In cancer cells, kind of that's not what we see. So we know that in cancer, there is chromosomal chaos. There are bits glued and stuck together here. There are duplications. There's like 4, 5. I mean, one of those is like, wow, it's really gone for it. Chromosomes are duplicated. Chromosomes are lost. Bits are stuck together. There's a phenomenon called chromothripsis, which is one of my favorite words. It means chromosome shattering, where we see whole chromosomes that have literally just smashed into pieces and been stitched back together again in any kind of way. We see all kinds of chromosomal aberrations. So this is not just looking for specific changes, specific mutations in specific genes. This is sort of a cellular catastrophe event that has happened. So again, when we're thinking about where does cancer come from, what causes it, it's what is that tipping point that turns a normal-ish cell, with some mutations here and there, but with normal-ish chromosomes. What is that trigger for this chromosomal chaos? And if we can really understand that, and maybe work out how do we detect these cells with aberrant chromosomes, and get rid of them, then again, that starts to become an interesting idea for how to prevent cancer. Because these duplicated chromosomes, this chromosomal catastrophe, this is rocket fuel for the evolution of cancer within the body. And this ultimately is what makes cancer so difficult to treat. Because cancer cells, they are genetically diverse. We know that cancers themselves are a patchwork of mutation. They are multiplying at speed. They are picking up more and more and more genetic changes. More chromosomal chaos is happening there. And within that crucible of evolution, or, bin fire of evolution, if you want to put it that way, there are all sorts of genetic changes that enable the cancer cells to spread through the body, to be resistant to almost any treatment that you could throw at it. And this is the fundamental problem that we have, particularly with advanced cancers. Once they've really got to this stage, once they are evolving, changing within the body, then we have a problem. And we are still not very good at treating these kinds of advanced cancers. And that's because our methodology, our idea, the modern idea for treating cancer is very much based on just finding the mutations in the genes and targeting them with drugs, the magic bullet idea of cancer therapy. And many of these very targeted drugs are the kinds of things about we read in the newspaper headlines, magic bullets, holy grails, wonder drugs, these drugs that cost incredible amounts of money. But when we actually look at what benefits do these very new, very expensive targeted therapies bring in terms of survival, it's often not that much. The benefits are measured in weeks, in months, sometimes in single-digit years. And this is a real problem because there's so much effort going into this kind of approach. Just find the mutations, develop the drugs, get it through clinical trials, just got to show a little bit more survival than the last drug that was like this. And there you go. It's not getting us to filling that glass to the top. It's putting more drops in. And there are some new treatments that are based on stimulating the immune system. These are drugs you may have heard of called immunotherapy. They don't work for everyone. They work for sort of fewer than 1 in 5 cancers that they are appropriate for. But they do seem to be quite exciting. And I think the immune system is a really exciting area of research. But there's an awful lot we need to know about these drugs and about the immune system to try and make more progress there. But I think that this approach of using very targeted therapies, it's a problem. And it's a problem for the reason that I just explained, which is that cancer evolves. So a demonstration of this is probably some of the most striking images in cancer research. I saw this presented at a cancer conference, I think, probably in about 2012, 2011. Yeah, it's 2011 when the paper came out. And this is a patient. This is a man who has malignant melanoma. So this is skin cancer that has spread throughout his body. And you can see where it's spread. This is the sort of the lumps and bumps throughout his body. And he was treated with a brand new drug at the time. This is a drug called vemurafenib, sometimes known by the brand name Zelboraf. And this is a drug that is very highly targeted to a specific genetic fault that we find in about 60% of malignant melanomas. It's a fault in a gene called BRAF. And we think that this overactive BRAF drives the melanoma cells to multiply. And so the idea, very obviously, you find a drug that blocks that faulty version of BRAF, stops the cancer cells from growing, the cancer is gone. And after 15 weeks of treatment, the person who was presenting this, he put up the slide and you could just hear this gasp in the room. This just looks incredible. You know, it's like it's a different person. This drug has been phenomenally successful in a matter of months. Absolutely incredible. And then it was the next picture that you could really hear the intake of breath. Because 8 weeks after that middle picture, all the cancer was back again. And this is the evolutionary problem, is that these drugs, they can be very successful. And it's not just particularly these drugs. We know of many cases where someone appears to be doing well on treatment. They're told, oh, the cancer's shrinking, and then it comes back. And it comes back because it has evolved resistance. Somewhere in there, in that maelstrom of evolution, of the patchwork of cells that are in there with all their genetic changes, there are cells that are resistant to almost any treatment that you could throw at it. And so what you're doing when you're treating cancers like that is effectively putting on a selective pressure, and you're getting rid of all the cells that are sensitive to the treatment, and you're leaving behind the ones that are resistant. And as soon as, you know, all those other cells are gone, the resistant ones start growing again. And in some cases, you may be lucky and there may be another drug that you can take, and you go through the cycle again. And when that one stops working, maybe there's another one you can take. And I call this approach the kind of the whack-a-mole approach to treatment. You have first-line therapy, second-line therapy, third-line therapy. But at some point, you're going to be out of options. And this is a real problem. And this is the heart sink, where I'm sure this has happened to all of us, where we know someone who's been treated, it all seems to be going well, and then the cancer has come back and there's nothing more that can be done. And that is, if you want to blame someone for it, blame Charles Darwin, because that is just evolution in action. Because once a cancer has got to a particular stage, there are going to be cells in there that are resistant to treatment. And you can treat that cancer, but you can't get rid of that pocket of resistance. And so that cancer is still there and it will come back. So what do we do? And I want to go back to the point I raised at the beginning, is that we do treat many cancers very successfully. And, you know, I don't often like to use the C word, the cure word, but there are cancers where we treat them and they do not come back in that person's lifetime. We can call that person cured. And we can certainly bring about long-term survival in many cases, that 50% of people surviving for at least 10 years after a diagnosis of cancer. But it is not enough. It is not good enough. And the more I sort of looked at many of the approaches, the ways that we're developing and testing new drugs, particularly these targeted therapies, I don't think that it's going to make a big impact. I think we need these drugs, because the more drugs we have, the more, and certainly looking in different spaces, trying to target different mechanisms in cancer cells. Cancer drug development has a really terrible Me Too problem, not that kind of Me Too, the sort of Me Too where every company is just going after a very narrow set of targets. Our cancer armament is basically just a bag of hammers, maybe a couple of spanners. We need a full toolkit. So we do need these drugs to be developed. But I really think we need to think about how do we use them in a much smarter way that takes into account the underlying evolutionary process that's going on. And the guy who I think is really doing this, my sort of personal cancer hero right now, is a man called Bob Gatenby. He's at the Moffitt Cancer Center in Tampa in Florida. And he is using his new weapon in the war on cancer is maths. And I'm terrible at maths. I just cannot do maths, can't understand it. And so it's always very distressing to me when I realize that so many things in biology are now coming down to maths. But, you know, there we are. But he and his colleagues are trying to use the mathematical biology, mathematical modeling, understanding the evolutionary processes and modeling them within cancer to come up with better ways of treating cancer. He's also one of the nicest people that I've ever met. We had lunch in his office, and then he let me, he's very interested in game theory and this idea of understanding the sort of the game between people treating cancer and the cancer itself. And so he let me chase him around his office doing a game theory game. So that's a really nice little bit in the book. So you can go and read that. But what is he trying to do? So he is kind of trying to come up with new ways of devising strategies to treat cancer that are different from the way that we currently tend to treat cancer today. So the way we currently tend to treat cancer today is through what we call the maximum tolerated dose, or MTD. And this is sort of exemplified by those pictures that I showed you of the man being treated with vemurafenib. So someone has cancer, they are treated with big doses of the drug. The idea is to shrink it down to nothing to make it go away. So you give a big dose, the tumor shrinks. You give a big dose, the tumor shrinks. You get to a point where, well, we can't see any more cancer cells. So, you know, good, yay, well done, it worked. And then the cancer comes back and it is resistant to the treatment. And that's because these big doses of the drug have got rid of all the sensitive cells in there, all the cells that can be killed by that drug. But of course, it can't touch the resistant cells because they are resistant to the drug. So you can keep putting that drug on and the resistant cells will keep growing. So over time, the cancer will come back and that drug will not work anymore. So Bob was sort of thinking about this. And he was thinking about new strategies. And he was reading an article about an agricultural pest called the diamondback moth. And he told me he was first drawn to this because it's a moth that eats cabbage and he absolutely hates cabbage. So he was very interested in understanding this moth. And in agriculture, this is the same problem that farmers deal with pests. But they sort of accept that, like, you know, if you treat your whole field with lots and lots of pesticide all the time and try and get rid of every single last pest, all you're going to do is encourage the growth of resistant pests, and then you are in real trouble. And he was like, well, that's exactly what we see in cancer. And I was like, well, so what do farmers do? You know, they've solved this problem because, you know, we still have crops. They're not completely overrun by resistant pests. And he realized that farmers were using a more sort of evolutionary approach where you accept that you have a certain level of pests in your field. That's just what you have to accept. And that within there will be some pests that are sensitive to the pesticide you use and some pests that are resistant. But those resistant pests are not as fit and healthy. They're just not as good as the sensitive ones. So normally you sort of want to balance these populations so that one is keeping the other under control and neither is getting out of hand. And so the sensitive bugs, they sort of grow up and you treat them, but down to a level where you're not trying to completely eradicate them, but trying to control them. And so he devised a strategy using his mathematical modeling, his evolutionary understanding, knowledge of the populations of cells, sensitive and resistant cells in cancers, to devise a clinical trial based on this idea. And it was a trial for prostate cancer. And this is how it works. So you accept that at the beginning, you have cells that are sensitive to your drug and you have cells that are resistant to your drug. And that resistant population is only a small population. So you could do genetic analysis. You could work out like what balance of cells do you have in that tumor? What are they likely to be sensitive or resistant to? And then you give them a dose of drug and you wait for the cancer to shrink, but only down to halfway. So you kind of accept that you're going to leave some cancer cells in the body and that some of those cells will be sensitive to the drug and some will be resistant to the drug. And then you stop treating. and you let the cancer grow again. And the cells that grow back are those sensitive cells. And they're kind of keeping the resistant cells in check. And then you treat again. And you shrink the cancer down halfway, and then you stop. And then the cancer grows again. And again, it's the sensitive cells that are coming back. And, you know, over time, that population of resistant cells does grow. But you're spinning this out for much longer, kind of riding this roller coaster. Or if you imagine like a rheostat, you're sort of balancing rather than just trying to turn it all the way down to zero, because that's just not going to work. And in prostate cancer, they had absolutely phenomenal results. So they were treating people with advanced prostate cancer with a drug called abiraterone, which is a sort of standard line therapy, sort of one of the later stage therapies for advanced prostate cancer. And normally men who are given this drug, from the point that you start taking it to the point where your cancer has just come back and it's resistant to the treatment, we call this time to progression, is about 18 months. And Bob, on his trial, he's had people surviving at least 4 years before their cancer has started to come back. Now, that is not a cure, but that is more than doubling what the current standard of treatment is. You know, if this was a new drug, people would be going nuts for it. There should be headlines everywhere talking about it's just using a drug that we have in a different way. Instead of trying to like drive it all down to zero, it's trying to balance these cell populations. There are trials in other cancers using this kind of adaptive approach underway. The key thing is you have to be able to monitor how much cancer is there. So with prostate cancer, you could do this with a test called the PSA test. It measures a chemical that's in the urine that's sort of a relative readout of how much cancer is in the body. So you need to have other ways of measuring how much cancer is there, whether that's through scans or blood tests. You kind of need that readout because you need to know when to stop and you need to know when to start again. And as I said, with this approach, eventually resistance does emerge, but it spins it out much, much longer. We need the results of more trials. We need bigger trials. You know, this is not the secret cure for cancer that everyone should be having, but I think this is absolutely incredibly exciting. It's about using drugs and treatments that we already have in a much smarter way based on evolution reality of what's going on. So we can think about this sort of approach as trying to control rather than cure cancer. And that's an interesting and challenging concept because we've been led by this idea of the cure. We all want the cure for cancer, you know, to get rid of it, to eradicate it, that it will never be there, never be there again. It's gone forever. We don't like this idea that you might have to live long term with it. It's difficult for us as a society when we've been fed this narrative of finding the cure for over 100 years. You know, since Cancer Research UK was founded in 1903, they want to find the cure. And the idea that the cure will just look like a treatment, you know, a bottle of pills that you buy that's labeled, you know, the cure for cancer. This is a different idea. It's about steering the evolution, the development of the disease within the body based on evolutionary reality and understanding the biology, understanding the genetics, bringing together the new genetic technology and tools that we have with mathematical biology, with evolutionary understanding in this sort of new way of thinking about cancer as an evolving process within the body. But as well as this idea of long-term control, we can start to think about, well, what does a cure look like from this evolutionary perspective? And that really is when you start to think about, rather than cure, you're talking about extinction. So we're very good as humans at driving populations to extinction. We have done it with many, many animal species all over the world. And this doesn't happen as just one kind of bonk, boom, that's it, that's gone. Extinctions happen through multiple processes acting on populations, driving them smaller and smaller to the point where they collapse. So as an example, Bob talks about the Heath Hen. So this is a Heath Hen. They were found all down the east coast of the US. And in the time of the colonial settlers, the settlers turned up and the colonists, they turned up and were like, woohoo, loads of these massive, big kind of chicken-like turkey things. Let's hunt them, eat them all. So there was a huge amount of hunting. And so by the 19th century, there was just a small pocket of these birds. They were living on Martha's Vineyard, the island off the east coast of America. And their habitat was shrinking as more and more people were living in the area. Habitats are shrinking down. So that's another thing. So you've got hunting is the big strike. You've got shrinking habitat. That's another strike. They get down to a small breeding population. There's not a lot of genetic diversity in there. And as I said earlier, the problem in cancer is you have huge genetic diversity in this sort of population of cells in the body. And you need genetic diversity in populations of animals to keep them going. You know, you've got to sort of mix and match it up to get out of the problems that you're finding yourself in. But when populations get very small, they have low genetic diversity. They don't have a lot of room for maneuver to evolve their way out of trouble. There was a fire on the breeding ground, a different type of threat. There were several harsh winters, a different type of threat. And then finally, there was a disease. And it just kind of did for them. The last Heath Hen died in 1932. It was so rare by that point. He had a name. He was called Booming Ben. And he was the last of his kind. They went extinct in 1932. So we know that kind of multiple different strikes at different times, at critical times, is enough to drive populations extinct. So how do we translate that idea into treating cancer? Well, we've actually already done it for some types of cancer. So the way that we now treat childhood leukemia, this is a huge success story in cancer research. So back in the 50s, a guy called Sidney Farber in Boston was trying to work out how to treat children with leukemia. And at the time, this was absolutely desperate. You know, virtually all children with leukemia died in a very short length of time. It was an incurable disease. And Sidney was determined to try and make a difference. And people said, you're wasting your time. Like, all these kids die. It's just no point. Like, what are you going to do? And he and his colleagues, with many rounds of clinical trials and, you know, to be honest, quite a lot of guesswork and trial and error, they came up with a strategy for treating children with leukemia that involves at least 4 different drugs with different mechanisms of action. They kind of work on completely different types of processes in cells, given at really specific times, drives these cancer populations to extinction. And now we have more than 90% of children with leukemia are cured. There's still issues because some of these children will go on to have lifetime side effects. So we certainly do need to work out how do we make these treatments kinder for children. But this approach of, you know, being more savvy about the right combination, the right time, how do we like finally bump that population of cells off? It's really sort of a much more rational, strategic way of using, in many cases, drugs that we already have in different ways to drive cancer populations to extinction. And I was talking to a researcher who works on malignant melanoma, and she was saying that she's starting to see some interesting results with some of the new immunotherapy drugs and surgery combined. It's like, it's actually, once the population of cancer cells is getting small enough, it just seems to fall apart. It just seems to go. So I think that there are some very interesting ideas coming about using all the different tools that we have, combinations, timing, using genetic techniques to monitor the different populations of cells in the body, and really being a lot more rational, based in evolutionary theory of what's going on in the body to treat cancer more effectively. And I think that's incredibly exciting. And at some point, though, we sort of have to say, well, you know, what are we driving this for? Because ultimately, you know, we all do have to die of something. I would very much like it not to be cancer. I think that for all of us who've lost people to cancer, it is not a nice way to go. It's terrible. It robs people of quality of life. It's a horrific, horrific disease. It will always be with us. There will be no way of preventing cancer from ever arising. As I said right at the beginning, it is the price we pay for being multicellular organisms that live in a world that is driven by evolution. So it's always going to be with us. This is always going to be a challenge. But my vision is by using these kind of treatment strategies these new ideas, really thinking about the underlying real biology of cancer and its evolution within the body, we can start to push that out and out and out and out for longer and longer and longer. And when I spoke to Peter Campbell, one of the researchers at the Sanger Institute, I said, will we ever cure cancer? What does a cure look like? And he was like, well, I guess it's that you die of something else first. So, I mean, that's not entirely a cheerful message to end on. But I'll end on a slightly more cheerful message, which was that cancer is a disease that causes us a lot of fear, and it causes us a lot of anxiety. For people who have cancer, for those of us who love people who are going through this, it's incredibly scary. And while writing this book, I have never been more attuned to my own mortality, every lump and bump in my body, everything, while I was writing this book. But genuinely, it has given me a new understanding of what this disease actually is. This is something that is intrinsic to our biology, that we need to learn to live with, to work with, and to try and steer to our own ends based on our understanding of what this disease really is. And I think in terms of the public understanding, this is not something external. It's not something that's modern. In many cases, it's not something that is anything to do with what you've done or anything in your life. This is an intrinsic biological process. We need to understand it, and we need to try and steer its evolution to our own ends. And I think if we can try and do that, then it will be something that we can understand more and that we can fear less. So that is my hope. I'll say thank you very much. Thanks to the Conway Hall team for having me. Please do buy the book. It's available now. I have a little website, rebelcellbook.com, and you can find all the links to various outlets and publishers, all good bookshops and the evil ones too. So thank you very, very much for your attention.
Yeah. That was a great talk. It was terrifying, illuminating, and humane. I think we're all about human solutions to human problems here, which is what you provided. The Q&A is now open. I know we've got one or two questions in already. Please raise your hand in the attendee section or put a question in the Q&A section to ask a question of Kat, and let us know if you'd like us to open your microphone. Kat, one of our team wanted to know whether blockchain is influencing the use of drugs within cancer.
I have thoughts about blockchain. I think blockchain is a solution in search of a problem. In terms of cancer, I haven't really come across blockchain type solutions there. I will say my sort of other life as a communicator of genetics, I do a regular podcast called Genetics Unzipped, where we sort of look at latest advances in genetics and all that kind of thing. I think there's possibly a use for blockchain in enabling people to own their own genetic data and make it accessible. And so potentially in terms of cancer research, you could imagine if people have, say, their medical notes or their medical information or their tumor sequencing and all that kind of thing, potentially that kind of way of enabling people to participate in research but maintaining ownership of their own data, I think is a possibly interesting thing. But yeah, I'm not massively a blockchain evangelist. I think in many cases it is a solution seeking a problem.
I don't completely know what it is, but
it's basically a spreadsheet that lives in the sky.
Okay, so it's like spreadsheet gold.
Yeah, it's spreadsheet god.
Okay, I can get down with that.
It's never wrong and it never changes.
Our next question is from Graham Taylor. Graham, your microphone's live. Are you there?
Yes, thank you. I'm here. Thank you very much, Scott. Can you hear me?
Yes.
Oh, great. Yeah, cheers. I'm Graham Taylor, a student at London Metropolitan University doing a Masters in Political Violence and Radicalisation. I really enjoyed your talk. It was brilliant, fantastic, really interesting, and fascinating to think just how easy it is to get cancer and how dangerous the actual disease is. It doesn't always come to mind. It's something that's always on our minds, I think, from general topic to general topic, about how we feel we're in danger of it, just in case it ever happens to us, but we always tend to ignore it and get on with our lives. I think that's the best ploy. my question to you is, with this test on prostate cancer you were talking about with the professor and everything, is the theory that we can constantly maintain the resistance to mutant cells over and over again? So hence, even if we can still have cancer, we can constantly treat it, and hence it'll be controlled for life.
Yeah, so that is the idea of this kind of approach, is that it's making it more of a controlled disease for longer. I will still say that in the current study that the resistant cells, they do still ultimately grow. So that is controlled for a limited number of years. I think that people investigating these kind of strategies are trying to work out how do you keep that roller coaster going and make it more of a chronic disease that is controlled for what would be your natural lifespan, rather than something that just seems to go downhill very fast once it's developed resistance to treatment. I will also say that you sort of started off by talking about the fear, the very natural fear that we all have about cancer. And one of the things writing this book made me realize that, yes, cancer is incredibly common, and we should all absolutely be aware of changes in our body. Anything that's not normal for you is definitely worth keeping an eye on, because the earlier anything is picked up, much, much more likely it is to be treated successfully. That is absolutely the case. But the other thing that I sort of realized was more of a healthy respect, that given how much mutation we all carry in our bodies, and that most of us will only develop cancer maybe once in our lifetime, it's like, my biology is amazing. My cells are doing amazing things to keep me healthy every single day. And it was more of an incredible respect for like how our tissues have evolved to keep us so healthy for so long, you know, that we don't get cancer all the time, given the kinds of assaults that our cells are under from all angles every single day. So it's given me much more of an appreciation and a respect for my own body. And it's like, yeah, I'm going to do what I can to like keep my tissue healthy and actually, you know, do what I can to not add to my mutational burden. Yeah, try not to do things that we know cause damage and genetic damage to DNA, things like, you know, smoking or too much exposure to sun, or sort of exposure to nasty environmental chemicals, but also to how do I keep my tissues healthy, you know, starting exercising more, drinking less, eating healthier, because I want to keep my tissues nice and healthy for as long as I can.
That's always a fear, and we tend to feel, you know, that fear is always prolonged on the mind, that even if we haven't got it and we don't look like we're going to get it, you always tend to fear the only slightest bump or slightest fall you might have on your skin, the tissue. You always tend to fear it. It's a horrible feeling, and we just all pray we don't get it, and I pray we never get it in our lifetimes.
Yeah, absolutely. And thanks for your question. Thank you.
Thank you, Kat.
Okay, we've got a question from anonymous attendee. We get a lot. They come onto a lot of events, so I'm going to have to read this one out. Forgive me, I'm not a doctor working in the field of cancer research, so there's a word I'm going to stumble on. There's been a lot of excitement around the use of oncolytic viruses.
Okay, yeah.
Are highly adaptable and targeted anti-cancer agents. Do you think the use of viruses will be a big part of cancer therapy in years to come?
Yeah, so this is an interesting idea, and I've been following the idea of oncolytic therapies for quite a while. So the idea is that these are viruses that are targeted to infect cancer cells, and then once they're in there, they kind of mess them up and kill them. And there's been some really interesting ideas. I think the main place where this is being researched is in China. Actually, there's quite a few trials going on in China. I know that Cancer Research UK was funding some work in this area. I don't know where that is now, because it's not sort of an area that I've particularly kept up with. But I think it's one of these things that's like, the idea seems really, really cool, and like it should really, really work. In practice, I think it has been very hard to actually make it work, to really make sure that you can get enough viral infection into every cancer cell in the body is a real challenge. And I think particularly now that we've seen the advent of immunotherapies, so these are therapies, the new generation of immunotherapies, kind of they unmask cancers to the immune system, or they kind of enable immune cells to recognize cancers. I think a lot of the people working in this idea of, you know, sort of viruses and immunity are now slightly more interested in that approach. Yeah, oncolytic viruses is an interesting one. I haven't seen amazing clinical trials. I know that there are some ongoing, So yeah, it's something that's interesting. And it's like all these things that are interesting. It's like, you know, the idea of adaptive therapy is like, it's really interesting. You've really got to prove that it works before everyone should run around and go like, yes, this is it. This is the thing. And work out who it works for and how it works and how do you deliver it safely and effectively. I think one of the problems with viruses actually is that we don't know in terms of like our own immune systems. And this is very tangentially an interesting challenge for the coronavirus vaccines, which are based on viruses, adenoviruses and things like that, harmless viruses that are engineered to elicit an immune response, is how much are our immune systems just good at getting rid of these viruses anyway? So that, I think, with virus-based vectors is an interesting challenge, like whether this wonderful vector that could get rid of your cancer cells, your immune system's just going to go, no, not having that, get rid of it. So that I think is an interesting challenge for those therapies.
Grumpy immune system. Our next question is David Quint. David, are you there?
Yes, I am.
Hello.
Hello.
Hi.
Yeah. Good to hear you.
What's your question?
Do you foresee any improvement for patients with mesothelioma?
Yeah, that is a very challenging cancer at the moment. I think, don't quote me on this. I'm not sure whether people are testing immunotherapies for it. I don't know exactly with mesothelioma where the progress is, but I certainly know that it has been a very, very challenging cancer. This is a lung cancer that's particularly caused by exposure to asbestos is one of the main causes of it. So yeah, I'm afraid I don't really know where things are with that at the moment. Sorry.
There is a report of one individual, a lady called Mavis Nye, who seems to have been successfully treated with Keytruda, but I think the experts are a bit skeptical.
Yeah, Keytruda is one of the. I think that's one of the immunotherapies.
Yes.
Yeah. So there's a really interesting thing generally with these immunotherapy drugs is that they seem to work better on cancers that have lots and lots and lots of mutations. That's because when your immune system is looking for cancer cells, looking for things that are wrong inside your body, it's looking for things that look weird. And because if cancer cells have lots and lots of genetic mutations inside them, they're making a lot of weird things and they look kind of freaky. They're also very good at hiding from the immune system as well. But generally, it looks like the immunotherapies that wake up the immune system to target cancers do work better on cancers that have lots of mutations and look kind of freaky. So that may well be an area. But yeah, with the immunotherapies, they don't work for everyone. They do seem to work. If they work, they work really well. And that's very interesting because it tells you the immune system is potentially a very, very powerful way to treat cancer if you can work out how to do it. But yeah, with mesothelioma, I don't currently know of any other things. But yeah, immunotherapy would be an interesting one there. Thank you.
Okay, we've got, I think, one more question from anonymous attendee, who is a more general question, I think. Is it true that some species, for example, naked mole rats, elephants, etc., are naturally highly resistant to cancer? And I'll give face there. And if so, are there any lessons to be learned from their biology that can help humans? Can we learn anything from the naked mole rat?
Oh, this is my favorite bit. So there's a whole big chunk in the beginning of the book that's looking exactly at this, about the different animals and how either are very resistant or very sensitive to developing cancer. And yeah, naked mole rats, it's sort of slightly a myth that naked mole rats never get cancer. There are described cases where naked mole rats have got cancer. But when we look at animals that do seem to be very resistant to cancer, these are the long-lived big animals. Naked mole rats are very small, but they're very long-lived and they're kind of very weird. They are eusocial. They live in a colony. They seem to have, they make this weird cellular glue that seems to keep their cells in place so they can't migrate around, which is a key thing in cancer. They also seem to have, their cells are they have a very tight sense of personal space. there's sort of a phenomenon called contact inhibition that protects us against cancer, where if your cells kind of start to feel that they're impinging on each other, they will die, because like clearly things are getting overcrowded. So those naked mole rat cells seem to be uniquely sensitive like that. Like as soon as they find another cell in their personal space, they're like, no, no, no. So that seems to be quite protective. Different species have solved this challenge in different ways. And there's something called Peto's paradox, which I talk about quite a lot in the book, which is Richard Peto, the epidemiologist, realized that in theory, really big animals, they're made of more cells, right? And their cells will multiply more and they live a very long time. So really big, long-lived animals should have loads of cancer, but weirdly they don't. And so there's a lot of interest in understanding this paradox, and that big animals, so elephants, they've solved the paradox by, they have lots and lots of copies of a gene called P53, which means that their cells are very sensitive when they get damaged. So if there's any genetic damage, the cells just die, poof, gone. So they can't become cancer cells. Bats, Brand's bats live for 40 years, tiny things, incredibly long-lived, incredibly cancer-resistant. They seem to have a mechanism that's to do with like sort of the caps on the ends of their chromosomes, these things called telomeres. Capybaras, very big rodents, live for a long time. They seem to have very good immune systems that mop up different damaged cells. And, oh God, I'm not sure how whales do it. I think people are really interested in whales, but you've got to get the whales to do the research on the whales, and that means you have to catch whales, and that's not great. So not sure about the whales, but yeah, lots of different species have solved this paradox in different ways. And yeah, there's definitely a lot of interest in learning from them. And I go into that in quite a lot of detail in the book. So buy the book if you'd like to know about that.
Buy the book. Thank you, Kat. That was, like I said before, that was illuminating and terrifying and humane and a bit hopeful. So thank you.
I hope so.
Thank you very much. Thank you for speaking to us.
Thank you very much for having me.
Thank you. We will be back again in the shed, not at Conway Hall, with various talks. Do look at our website, conwayhall.org.uk, to see what else we're doing. All manner of events, and we hope you have a good rest of the weekend and next week. Here's hoping we don't get locked down soon. Thank you very much, everyone. Thanks again, Kat. Good.